get coming here and talk to you. These are the people who actually do the work. They are the people in the lab that actually put in their hard work, making the molecule and the nanoparticle that I'm going to tell you about today. I also collaborated with these faculty members in the medical school, Northwestern University, have two campuses. We have a campus down in Chicago in the Midwest of the US that is a medical school, and they have a lot of very talented faculty members that have an animal model that we make our particle and we go down there. It's 1 hour between the two campuses because of the traffic jam. And, we take our material down there and they do the mouse study, and sometimes they will get into animal study as well. Okay, so the idea of cancer chemotherapy is actually a very obnoxious idea to begin with. First of all, you have a patient that have cancer, which is a fast-growing organism. And what you want to do is you want to give the cancer a drug that is a poison. And you want that poison to kill the cancer cell before it kills the patient. And most doctors don't know how to do it very well because they fail organic chemistry. They don't know how to do titration. And so the challenge with that is that you ended up having a lot of side effects, right? And side effects ranging from losing hair or vomiting, or also have a compromised immune system that require the patient to be isolated. And these are very devastating. If you ever have a friend or relative who went through cancer chemotherapy, you see a lot of this happening. So what we want to do is we want to see whether we can target the drug that are used for cancer chemotherapy to the particular disease tissue. So these are cancer cell, can be metastatic cancer cell, or can be a cancer, a solid cancer in many cases. And what you want to do is you want to package the drug into a small nanoparticle that would then go directly to the disease site and then release the drug right there, right? And that would hopefully reduce the side effect that I just mentioned in a couple of slides before. And the other pathway that potentially would be really interesting is that you can combine the drug together with an imaging agent. And that imaging agent would allow the monitoring of whether the cancer actually drop in size or not. So if you have a solid cancer, can it actually decrease in size as you gave the treatment? Then this right here is the targeting group. It could be an antibody that allow the particle to actually go to the cancer site a lot easier. And then the last bit, that because of this delivery, you might be able to repurpose a lot of the drugs that are normally not available for cancer chemotherapy and open up new mechanisms that would treat cancer. And that's the last part of the talk that I will mention to you here, that this is a second platform that we talk about, okay? And then in terms of affordability, if you can also have these nanoparticle packaging, you may be able to give the particle to the patient once and then reduce the number of visits so that you don't have to go to the doctor and get the chemotherapy very often, because these particles can have an ability to release the drug over a long period of time. Okay, so with that, I want to show you a picture as a bio-inspiration motivation of what we had. So basically, this is a viral particle, and after the pandemic, hopefully everybody is very familiar with the structure of the virus. The virus have a payload here, and the payload have a protein coating that actually stabilize that payload. And on the surface of the virus, you have these recognition group, right? These are epitope that would go to the right tissues. In the flu or cold, it go to your throat tissue. It's actually start infecting there. And once it binds to the tissue, it will open up, this protective coating would open up and release the payload. And that payload, viral DNA, RNA, that would go into the particle, go into your cell and break up and actually make more viral particle, and that's how you get sick, right? So this is a structure that is very amazing because it's the best way to develop the a protection for the payload. And it's biocompatible because it can't get into the body. And really the key here are spatial and temporal recognition. It needs to go to the right place, and it needs to then release the payload at the same time. So what we want to do in our nanoparticle is try to mimic this design principle in order for us to actually make the particle. So these are the requirements that over 20 years of work in the field. So what we want the delivery vehicle to be assembled from off-the-shelf component. If you want it to be easily accessible and affordable to everybody, it has to be off the shelf. It should not be very expensive, right? And it should be small enough. So in the olden day, people say that 400 nanometer. And nowadays, in 2014, you want it to be between 30 and 70 nanometer, right? And there are a lot of reasons for that, but for now, you can just accept that. And then it should be stable and biological inert, and it should release only by interaction with the disease tissue. And the last bit that most people don't really talk about, that it should be biodegradable. So once you release the drug, nothing should remain behind that actually cause harm. And that is a lot of the time is not a part of the design in some of the current system. So I want to show you a system that was a kind of an inspiration of our work. So this is a real drug that in the US that was invented about the early 2000s. This is called Doxil. What it is it's take this drug right here. That's doxorubicin. That's a DNA intercalation agent. And they put it inside the liposome. And this is the lipid bilayer, for those of you who still remember your organic chemistry. It really are just these long tail lipids that have a head group that actually make kind of a soapy-like layer, and that isolated the inside from the outside. So the inside here is aqueous and the outside here is aqueous, right? And this is the liposome. And so when people make this doxil drug, it is interesting because what they choose is they choose this medium size nanoparticle. It is in the sand, this 100 to 120 nanometer particle is not too stable that it would keep the drug payload inside. And it's not too unstable that it would release the drug as it go through the body. So they pick this middle Goldilocks range, for example. And here's shown here as an example of some of these liposomes if you leave them too long. So these are the tiny liposomes that you make, and this is an electron microscope. And if you don't make it right, what happens is that these little soap bubbles will combine, and you can see them merging together like this and make the bigger particle. And this is the challenge with these liposome-based structure delivery, is that if you don't have a protecting mechanism, they will combine and make big particle, and that's bad. Because when they make these big particles, they combine and they fuse and they also leakage some of the drug outside. And that drug can cause side effects. So what we want to do is we want to actually try to stabilize this by going to the next generation of design. And what we do here is we take a liposome that's loaded with a drug, and then we would insert a polymer, and this polymer is a 5-step synthesis that you can make in gram quantity. And you insert this in here. This end right here is a cholesterol molecule. So here's the cholesterol that go into the lipid bilayer. So you insert this into the lipid bilayer membrane. This is called polymer grafted liposome. And then you cross-link these tether together and you make a net. So essentially you make a net around this. And this is the net that actually protecting the particle from combining together. Right? And this is done by my student, Sang-Ming Lee, who's now a professor in Korea. And we call these things a polymer cage liposome because you can load any kind of drug in here. And I will show you a lot of examples of that later. And what this net does is not only protecting the drug payload, it also allows the particle to release the drug. And this is a very interesting physiological mechanism. So you have these polymers that crisscrossing the surface of the particle. And when it gets protonated, because it goes into a cancer cell that is very acidic environment, cancer cells are typically hypoxic, it will actually cause hydrogen bond at these linkages here and it shrink. And so the cartoon idea, and we actually have a modeling on the computer to show that this is a reasonable mechanism. So you have this polymer network on the surface of the particle like that. When it go into the cancer cell and it see acidic environment, it collapses. And when it collapses, the net would just create pressure inward and then release the drug. In some ways, very much similar to the heat shrink tubing that you use in engineering, or you have a sponge that actually collapses based on the salt condition or temperature, okay? So here's an example of how that release mechanism works. So what we have here, we monitor the diameter of the particle. It's a function of time, and this is the log scale here. We have pH 7.4 to begin with for 10 minutes, and then we drop the pH. And what you see here, this is our particle with the network, okay? And you see how it collapsed, and actually by interpretation, it released the payload. And if you have just a polymer grafted liposome, it doesn't do anything. It just stays flat. And then you have the uncrossing polymer, or actually the plain particle, it actually also stays flat. And so this is the idea that it actually know where the particle go into the acidity of the cancer tissue, and that's how it collapses. Okay, so let me just show you over the last 2 decades or so, what we have been able to do is we have able to load a lot of different type of drug in here. We load inorganic drug. These are arsenic oxide. These are rat poison, but it's actually used for treatment of leukemia. Doxorubicin that I just showed you a while ago. And then the A-chloroadenosine and A-aminoadenosine, these are the ones that actually are nucleotide that specifically for treatment of leukemia. And so we was loading all these things with fairly high drug to lipid ratio. And we was able to put a fluorophore imaging here, as well as MRI imaging agent. We put antibody in here. And that's the general platform that we have used for a lot of our drug delivery. And so I want to show you here one of the first mouse study that we did. This is a mouse with cancer that actually on the thigh of the mouse. And we inject it into the peritoneal cavity. And what we do here, we do 4 injections over 3 weeks. And then we monitor the volume of the tumor, how fast the tumor grow over the next 3 weeks. And what you see here, this is our particle, the red one. You see that it was able to keep the tumor grow down comparing to the free drug. So the free drug got injected and it does keep it down, but then it metabolized and get resistant, so the cancer go up. And here is the control. This is the free particle that have nothing in it. And so clearly that the particle have an effect, okay? And here you show that these are imaging that I was mentioning to you. This is our control mice, and these are the ones with the empty vessel. So you see the tumor are very big, and the redder it is, that means the tumor very intense. This is the free drug. After the 3 weeks, again, you see the frog get metabolized, and it's actually very red. And these are the mouse that injected our particle. On average, it look a lot red, less red, and that means that it actually does reduce the mouse tumor weight. And now what we can do with this is we can go to very high dosage. So the image before that I showed you was 2 milligrams per kilogram. And now we increase to about 7.5 milligrams per kilogram of the drug. And this is actually in normal dose, it would kill all the mice. But in our particle, you don't see that. You actually see the mouse gaining weight, as happy as the mouse can be when they got cancer. And none of these die, right? And in fact, in here, we can actually keep the tumor down comparing to the dosage. So the lowest dosage, you actually see the decrease less than the higher dosage. So this is actually good. We go in the right direction, except that we have only a single drug, right? And for most cancer chemotherapy, a single drug doesn't work. So what we need to do is we need to go to double drug. And I will show you that in the next slide here. So in the next slide, what we want to do is we want to actually take this drug right here that we loaded, and now we put an imaging agent and a targeting group, okay? And this is the one that targeting breast cancer. And I want to show you that if you actually use a cancer cell that have a lot of receptor on the surface, and you have these antibody that targeting, we will see that it actually dropped the effective dose. So you're now using less drug comparing to the one particle that doesn't have the receptor. And so now what we're going to do is we're going to go ready to go with the second drug. So shown here as an example, now we have a second drug where we have the drug that is inside here, and now we have on the outside, we have also a biologic. These are the DNA that would also target another mechanism. And what we do here is inject directly into the mouse brain. And where we see right here, this is the brain of the mouse that is not treated. This is the brain of the mouse that are treated with our particle. You see right here, the tumor is very red and big. It's actually fill up the whole head of the mouse. And here the tumor get reduced. And so we have to kill the mouse before, but it looked very promising that we should be able to actually reducing the burden of this tumor there. So we're still going on with this work. And what we really want to do now to make it more effective is reduce the size. So what the issue with 100 nanometer is that it doesn't go through the blood-brain barrier very well. So now we have to reduce it down to 25 nanometer. And then we put the biologic on the surface and then the molecular drug in the inside. And that's what we really want to do here. This is the combination that we think if we eventually got to the point where it can treat the glioblastoma, this particular tumor that we mentioned. Okay. So that's finished the first platform that I mentioned to you. Now I'm going to switch over to a second platform for a few slides just to show you the repurposing of the drug using this platform. So this molecule here is a commercial cream that it used to treat vitiligo. So those of you who are familiar with Michael Jackson, the singer, you know that he bleached out his skin, and he bleaches out his skin using this cream right here. He basically killed all the pigment cells on his skin and make it not working, so his skin become white. So that is an abnormal use of this drug. This drug turned out that can kill the cancer cell that due to the malfunction of those pigment cells. Okay. The problem is that it's not very soluble and it's highly toxic. So if we really want to use it internally instead of the cream, where if you have metastatic melanoma that go everywhere, you want this drug to actually be able to go inside the body and go to the cancer side. And that's what we're going to try to do here. So before I show you how we do this, I'm going to show you the example of how this drug kills these pigment cells. So these are the pigment cells on the mouse skin, and you can see these are dark spots right here. So if you actually treat that the skin right there with this drug as a cream, you see that it eats away all the pigment cells. So it turns the skin white. But the other thing that is interesting about this drug is that it can recruiting immune cell to go to the cancer. So this is the control skin. This is the skin that we treat with that drug. You see all these bright dots? These are where the immune cell in the body is recruited over to kill the cancer cell. So this have a two-pronged mechanism that is discovered by my colleague, Dr. Caroline LaPoule, that it's a single molecule, but it kills both cancer cells as well as recruiting immune cells to kill the cancer. So it's a two-pronged mechanism and it turned out to be really inexpensive. This molecule is 100 grams for like $1. So you can if you can figure out how to deliver it to the patient, it will be very affordable. So that's what we set out to do. Okay. We take this molecule, and by organic chemistry, a two-step synthesis, we attach a long tail to this, and then we insert it into the lipid bilayer. And this is the whole plan that we want to do. We're going to insert it into here, and we're going to actually put a polymer layer that I tell you on top, and then we're going to attach the antibody on there. And that's going to make it really targeting, okay? And before we do all of that work, we want to test and see whether the idea actually accomplished by just adding a long tail into the molecule, because sometimes you have molecule and you change a little bit, it doesn't quite work. So what we do is we take this molecule and we just add a long tail to it, and then we add an imaging lipid in here just so that we can see where the particle goes. And this is a picture of the cell on the mouse skin. This is the normal mouse skin, and these are the melanoma mouse cell. And we expose this for essentially 60 minutes. So we just take this particle and we rub it on the mouse skin for 60 minutes. And then here we just inject it into the melanoma cell. And what you see here is that the mouse skin doesn't really fluoresce. That means that the particle doesn't go into the mouse skin cell. But over here, you see that there's a twofold increase within 60 minutes of the melanoma cell uptake the particle. So our particle is now very specific, or at least more specific for the tumor, that it uptakes a lot more of the particle. And then I will show you here that it reduced the dosage too. So this is the free drug. So in culture cell, you see the IC50 of the drug here is about 250 micromolar. But when you go to our particle, you now can drop it down to roughly 10 to 25 nanomolar. And this is actually really promising because you can reduce the dosage. Okay, so now if you increase the dosage, you see that basically this is the control, and this is the one where we treat with the particle. You kill all the cell right here. And in fact, some of the formulation we had completely killed the cell. You know, out of, this is like 100 micromolar compared to 900 micromolar here. So we have a really promising drug. And what we are doing is we're forming a company that's trying to scale this up and actually make it available for clinical trial. We think that this would actually be a complement with between standard care right now for metastatic melanoma. These are really expensive treatment that TKI or the anti PD-1 and anti CTLA-4 checkpoint inhibitor. They're really expensive, and it should be make really cheap because the molecule is cheap, and you can use it for any of the melanoma patient at any stage. It'll be combining with other treatment. And we think that it actually can be deployed for patient segment that have a resistance because of the mechanism is different. And ideally, this can be delivered to any patient by any clinic, like local clinic with nurses and doctor that just know how to inject into the patient. And we estimate that if we are successful, we can treat initially at about 8,000 American, and eventually, once we get enough data here, we can probably got to a group of people, about 100,000 people worldwide. And this is about a quarter of the near half a million cases every year, worldwide melanoma cases. Okay. So in conclusion, I want to tell you that synthetic chemistry, you can innovate a lot of the synthetic chemistry that combine with material so that we can really make the particle targeted, therefore make it personal to the patient by reducing the dosage, to make sure that it go only to the dry cell. And then also the other part is that we can repurpose this and expand the scope of some of the current drug to make it more efficacious. And with that, I assume that I will answer the question at some point during this symposium. Thank you.
Thank you, Professor Nguyen, for these two innovative and affordable treatment of cancer. Please keep your question, and we invite the second speaker to come. Let's say, Maher. Maher, you have the floor. Introduce yourself, please.
Yeah, Mahem is Senegalese. What she's doing is just amazing. She had her A level, A level, c'est le baccalauréat, in 2016, and now she is based in Netherlands, and you will working on stem cells, and you will see how great she is, and just amazing. After Mahem, we will have myself. So as I told you, medical doctor and training and in London. And we will have also Salif from Ivory Coast. Mahem, are you done?
We have the it's fine. I'll use my computer. Okay. Hi. Thank you. Thank you for the presentation. And most importantly, thank you to the organizers for having invited me to this conference. As Dr. Cisse said, I actually grew up in Senegal. I studied in Senegal and I left about 7 years ago to pursue my studies. So it's really a pleasure for me to be able to come and present and show what I've been doing outside from Senegal all along. So I'm a PhD candidate in the Netherlands, in Utrecht. Specifically, it's a small city half an hour away from Amsterdam. And I'm in my 3rd year of my PhD, actually. And I work in a field called biofabrication. I will explain what it is. And most specifically, within biofabrication, I do 3D bioprinting. And I will just showcase, hopefully. Yeah. Okay. So I will not exactly dive into the specifics of my research, but show more how this field can help in the field of personalized medicine in general. So I won't need this. So what is biofabrication? So biofabrication is a field that en fait, je peux pas faire si j'ai pas. On voit juste un peu. Oh. Okay. So what is biofabrication? So it's a field, so in general, I work in a field called tissue engineering, and the overarching goal of the field of tissue engineering is to be able to create organs in the lab in a more general manner, because whether it's the liver, the heart, you always need donors, and you don't have enough donors for the amount of patients they need. And across time, we figured out that what we try to do in the lab is very simplistic. So we just put cells in a culture medium, but that does not mimic what happens in the body, because there are so many players that exist that we never take into account. So that's how tissue engineering and then biofabrication came to be. So biofabrication is the mix between using cells, using biomaterials, and using integrative cues to be able to build organs in a 3-dimensional manner. Within biofabrication, at some point, we figured out that just putting all of these 3 things together was not enough to build organs with with a good spatial resolution. So you might be able to mix cells together, but you would not be able to put them where you want them or a specific location. So that's how 3D printing came to be. So 3D printing started usually with plastic or with metal. You have a syringe and then you put your material in it and you're able to build very fast layer by layer structures in 3D. And it was a revolution in a lot of different fields. And within 3D printing, people thought, what if we put cells and biomaterials instead of just plastic and metal? And can we create organs that have a physiological scale that way? And that would start with having a model, a 3D model on your computer. So let's say, for example, you want to print a heart. You can use the MRI of a heart. You can put it on your computer and have you can put it on your computer and have the 3D model on your computer, send it to then the machine and have the machine build the heart out of the material that you've chosen. Of course, that is very far in the future because there's a lot of things that we have not figured out yet. What cells to use, how to use them, where to put them, a lot of different things. But what I want to show you is what are the advances that have been done at the moment and what we can build actually, what type of organs we can get to using different 3D printing techniques. And for that, I have to say, without visual, it's going to be very hard. So I'm just going to wait for a second. If you have already any questions on tissue engineering, now's the time to ask them, maybe. Okay, I sent the link. Oh, yeah. Yeah, then it's also Okay, I'm back. So if you remember merci, from 5 minutes ago, this is what I was explaining. So tissue engineering is a combination of all these three, and then with 3D printing, you're able to build larger scale organs with more precise depositions. So how does it work? Let's imagine, so you have an MRI or a scan of whatever organ you'd like to print. You send it to your computer, and the computer will basically cut it into parts that then it will be able to understand. It will transform it into a mesh, and then it will convert it to a file that the computer will understand and that it will be able to print brick by brick. So there are a lot of different printing techniques available. I will briefly go through some of them that are more commonly used. So this is the one that I was explaining at first, which is the more traditional one that was used with bioprinting. You can put some cells in a needle like this, and then you extrude it on a receiver plate, and then you can build whatever organ you'd wish like in 3D. However, with this technique, we saw that there were very long printing times to get to a certain size, because by the time that everything happens, most your cells die, and there's also a lot of shear stress that happens in here because your cells are really pushed through. So researchers came with the idea of using light to polymerize gels at specific locations and increase the spatial temporal resolution of the prints. And that's how different light-based bioprinting techniques came to be. I will not explain all of them. Of course, if you want more details, please, I'll be here until Thursday. Come to me and we can discuss several of these techniques. But what I will be talking about later on will be volumetric bioprinting, which is what I work with in my PhD. So the way volumetric I'll explain later how volumetric bioprinting works. But to show you a couple of applications on how this could be used for personalized medicine, one of them is this printing technique called laser-induced forward transfer. So very briefly, you have a laser pulse that is here that will be sent into a plate here, a slide that has cells and a biomaterial and some cells. And with the strength of the laser, the cells will be shot down on what we call a donor plate or receiver plate. And you can place cells wherever you want on this receiver plate to build the organ that you wish to use. So this printer was created by a company called Poieris. It is located in France, in Bordeaux. And they are working on a skin substitute called the Poieskin that you see here. So with this printing technique, they were able to actually create the different stratified layers of skin. And the idea of this printer is that they would like to use it as a therapeutic product for chronic wound healing of lesions such as diabetic foot ulcers. And what is very interesting with this printer is that it was the first printer that was placed in a GMP environment, which is an environment that is that has more regulations that can be used for healthcare. And it was placed in the EPHP hospital of Marseille. So it's very, it's really a revolution, because you can have the printer at the site where the doctors actually need it, and at the site where you can train people to use it for different purposes. You can also build, so this is a work that is being done with some colleagues in the lab where I work at. So the idea of bioprinting is that it was first optimized for hard structures such as the bone and the cartilage, because those are the structures that were more simple in terms of complexity, but also the structures that were easier to print, because the material that you need for that, it's hard. You can use metal, you can use ceramic, so you don't need to dive too deep into the complexity of the material. So in the lab where I work right now, they're trying to basically use these printing techniques to generate a patient-specific osteochondral plug, which is the part of the bone that you see here, and it's a location where you can have a lot of orthopedics defect, and it's very hard to actually replace that orthopedics defect. At the moment, most of the therapeutics available are based on materials such as ceramics, but those have their own disadvantages. So they use a technique called melt electrowriting, and with this technique, they're able to deposit a filament, and they can deposit the filament that has the shape of the exact location where the defect is. So you can have the scan of the defect previously done to this and then print exactly what has to be placed there. And here you have the example that was done by one of my colleagues, and she just shows with these different immunostainings that you can see here that when you place cells in those structures, they actually differentiate into the tissues you want them to be. So they can differentiate into both bone and cartilage. And even further, they can have the architectural structure that cartilage, for example, has in vivo, because it is a very architectural organ that you have. And then one last example, which is closer to what I actually do in the lab. So this is a smaller device, which is a microfluidic chip that could be used for printing of a renal proximal tumor model. And this would be very interesting in the context of drug testing and drug discovery, and also disease modeling, because you can seed patient-specific cells into these structures that are 3D printed and be able to very fast screen drugs, because of course the drug testing, the drug discovery pipeline uses a lot of money on models that are usually 2-dimensional, and it actually lose some of the 3-dimensional complexity you need to accurately be able to predict the behavior of a drug. Sorry, no worries. So I will try to go a bit faster maybe in this last part, because I think we lost a bit of time. So let me know if I miss you or if something is not clear enough later on. But one thing that I have not mentioned yet is the cell source that we use to print all of these organs. So usually people use single cells that could be primary cells that are very easy to get depending on the organ, but depending on the cell, they have a limited repopulation capacity, and you can only use a certain number for a certain amount of time. So you always need people to actually donate cells to be able to use it in the lab. And some years ago, in 2009, some researchers Utrecht discovered what we call organoids. So organoids are what we call mini organs. They're not exactly mini organs, but they're stem cell structures that very accurately mimic the physiological organ from which they are derived from. You can derive them from virtually everything at the moment, from healthy tissue, from diseased tissue, from tumors, from the heart, the brain, anything. And it's a very nice structure to use, because there's a lot of things that are present in organoids that you don't have with single cells. Since they are these 3-dimensional structures, they already have all the interactions between the cells that you need to then form the organ later on. So you bypass the step that you need to build those interactions by your own, because these organoids do them on their own. So part of my project is to actually combine the stem cell work with organoids with 3D bioprinting to be able to build physiological size organs that could mimic even closer what we would need to be able to transplant them or replace some of them in the body. I will pass through this. And with this last part, volumetric bioprinting, which is the core focus of my work. So I was explaining before that light-based printing technologies have really enabled people to get a higher spatial-temporal resolution. However, one of the drawbacks of light-based printing techniques is that the size was very diminished compared to what you could get with extrusion-based printing, for example, with nozzles. So in 2019, some researchers figured out that they could harness the power of the CT scan, but in reverse. So when you do a CT scan, you have a machine that kind of takes 2D planes of your body in different angles, and then you have a mathematical model that reconstructs it, and you have a 3D vision of whatever you just scanned. We do the same thing, but in opposite. So we have a 3D object that we want to build. We will cut it in different 2D planes, and we will project those lights into this device called a digital micromirror device, and through a series of different lenses. So this is all done by a colleague of mine. I don't have the specific optics expertise, but it goes through a lot of lenses and it gets to a rotating stage that has a photoresin inside. And this is a real life video of what happens basically in this rotating vial. You have here just a ball, an empty ball that we printed that appears in seconds. So this was actually a revolution because it was very fast and you could get to very big structures with no drawbacks on the cells. So the cells would not stay in a biomaterial that they don't like for long. One of the main drawbacks of this technique, however, is that we have to use photosensitive biomaterials because we use a laser that we shine on the material to cross-link it. And at the moment, I'm trying to work on identifying how the light affects those cells. And oh yeah, this is a video that shows the process. So you have the vial that you insert in this little device here. And I don't know if it's easy to see. And you have a projection that will be shown on the vial on the rotating stage right now. Exactly. So this is a pancreas, for example, because some colleagues of mine are trying to 3D print a pancreas for drug testing purposes. And within seconds, you can see the pancreas appear on the screen as it is printed, as it is being polymerized. And then you can just take out the vial with your print, wash it, and then flush out your print. So as we add water to the uncross-linked resin, you will see the pancreas appear. And that is what we're using right now as a model of organ modeling. And this is a study that a colleague of mine did a year ago, where we actually used this technique to print liver organoids. So as I was mentioning before, organoids are mini organ structures. And we were trying to see if using this printing process, we could create intricate architectures to study liver toxicity. Because of course, in the drug testing pipeline, one major issue is that you might have a drug that will have its intended effects on a specific cell type, but then it will fail all the phases of the clinical trials because it will have an attended liver toxicity. And we saw that when we put liver organoids into these printed structures that we perfused with urea, we could see that the liver actually metabolized it. So it actually functioned the way it would in vivo. This, of course, is a very preliminary steps. There's a lot of things to do before being able to bring this to the clinic, but that's what we aim to achieve at the moment. And all of these things have to be done together to be able to bring this research forward. So not only do we have to look at the bioprinting techniques, but also what type of bioinks we're using. As I was mentioning, we use light. So we have to make sure that the cells are okay with light, that there's no DNA damage, that there's nothing happening to them to bring them to clinical trials. And to finish this talk, this is something that is quite important for me to mention because something that I've tried to do for the past couple of weeks is I've just been trying to think about how my research, how the knowledge that I've been acquiring in Europe could actually help people here. Because of course I do bioprinting, it's very cool and all, but in the real state of things, it will not be helpful in the field for the moment. So I've been in contact with people such as the people who created this Ubora platform, which is the biomedical engineering e-platform for innovation through education. So this was created by Italian researchers from the University of Pisa. They received grants from the Horizon 2020, which is an European EU funding. And they're in collaboration with the UNECA, which is the United Nations Commissions for Economics in Africa. And they basically created a platform where researchers in Italy could organize competitions or design competitions, workshops to show people how they could use 3D printing and biofabrication actually in countries that have less means that you would have in Europe. One drawback that the person who created this told me is that for now they could only do it in Eastern African countries such as Kenya or Ethiopia because they are English-speaking countries, and they can't yet do it in West Africa because they don't have French speakers. They're from Italy. So one also goal of me to be here was to be able to actually know what people on the field would need in terms of equipment, in terms of design, because I was explaining before to some people that 3D printers look very fancy, but they're very easy to build, and you can print a lot of things that you would need in the lab for not a lot of money. So I think it's a very interesting platform and a very interesting thing that exists, that it would be quite beneficial to also have it in French-speaking countries. And with that, it's the end of my presentation. This is my team in Utrecht, and thank you for your attention, everyone.
Merci pour cette présentation des applications du bisprinting, comme vous dites. Donc, vous allez garder les questions à poser à Marem. En attendant, nous allons suivre la présentation du Dr Badara Cissé. Il va nous faire un peu le panorama de la médecine personnalisée en Afrique. Vous avez la parole.
So my talk will be about personalized medicine, a concept that is still very poorly known in Africa. And here at IOSF, we are part of a consortium, Europe, Africa, personalized medicine. We are at the last year of the consortium, and I will talk you about the cartography. We did a mapping of personalized medicine in Africa. Okay, so I will go fast. So we need to explore new model in our countries in Africa to tackle the high morbidity, mortality. And one of these models can be personalized medicine. Personalized medicine is just about profiling the patient characteristic using the phenotype and genotype of the patient for disease surveillance, prevention, treatment, etc. So, as I told you, the concept is really poorly known, and we have to make an effort for making the concept a little bit better known. In North Africa, Egypt, and in South Africa, personalized medicine is just, the momentum is just quite high. There are people who call personalized medicine the medicine of omics or the medicine of the future. But in our countries, lack of infrastructure or equipment, not a lot of specialists, limited experience of those who are dealing in it, isolated collaboration. We are not used to working together in Africa. Instead, we are even competing sometimes. And you were talking, Salif, about regular, I think that was you or someone from in the plenary about regulatory bodies and ethics committee needing to be upgraded and standardized. So we did in the Europe Africa Permit Consortium, we did a mapping of, if you take genomic research, we made a classification of 5 groups depending on the level of performance. You can see that here in South Africa and in Egypt, these country scientists are doing very well. There is a high level of health research governance, financing of research, human resources, the output, and international collaboration. They are doing quite well. And those at level 5 are the group that the performance in genomic are low. So we try, based on this 5 categories. We try to classify each of these countries. We also try to classify the countries based on the level of publication. You will see that here in Senegal, Senegal was halfway through the table, but for the 2 past years, we create a lab of genomics here in this institute, and you will see that we have had a lot of publication just over 24 months. This one in Nature, many others, 15, I think, in total. And even last month, November 15, there is an outbreak of chikungunya in Kédougou, in the southeast of the country. And Abdou Padan and his team were able to detect the virus and prove that the virus, that was a new lineage, a new clade of the chikungunya, never reported before. And the publication was immediately made. And this is just, maybe you are among the first to know that. So, another observation was that this new clade was very strongly related to climate change. So I have to say, I made a survey, and I approached 82 champion or champion to be all over West and Central Africa, focusing on West and Central Africa. And I received 35 responses, once again, in countries of West and Central Africa only, and asking them a couple of questions on personalized medicine. And then we had a virtual workshop to discuss the results. So most of them I have to tell you, and all the results of the survey are in this link that I can share with you. So we did a SWOT analysis, trying to understand, for instance, what are the weaknesses for developing personalized medicine. And once again, personalized medicine is the medicine of the future. We have to take these steps. If not, we will be in trouble in the future. So, so you will see, I think there is no need to go through all of them. But just if for the weaknesses, the concept not known, we need to increase awareness. The governance of the research is not favorable to developing personalized medicine. And if we take the genomic lab, there are still many countries in West and Central Africa without a genomic lab. And when we discuss with the champion or champion to be, even in the same country, they can have contradictory results. So some thinking that there is a genomic lab, other thinking that they are not. So just wanted to say that personalized medicine, we have to do something. And we are working with Professor Anta. We are working on developing a consortium for West and Central Africa. We were in Nairobi last time. We had a meeting there. And we are hoping to be able to organize a workshop next June for the first step of that. We have already written the concept note for developing a consortium for West and Central Africa. But in this region, we are lagging behind. North Africa, South Africa, and the rest of the world. They are making real progress. Kenya in particular, but we have to make an effort. We have definitely to make an effort. Thank you.
Thank you. Merci, Badara, pour ce panorama de la médecine personnalisée en Afrique. Merci beaucoup. Donc, nous allons terminer par la dernière présentation, celle du docteur Salif Djino, pour cette session sur la médecine personnalisée.
Okay, thank you. I'm Dr. Salif Djino. I'm medical lead for Roche Diagnostic based in Ivory Coast. I'm very happy to be here to share what we are Roche, we are doing for personal healthcare in Africa. So because of the time, I will not go deep into what is personal healthcare. I will just share with you an overview of personal healthcare, the meaning of that within Roche, and what are the different initiatives we have started to make it happen in Africa. Because for us, Africa should lead this new approach. Something has been doing, as Professor Cisse said, in Europe, so that we think that Africa should not miss this new journey to treat the patients. So for us, within Roche, this is what we are thinking. So the health system now is not only in the low and middle income countries, it's everywhere. The needs are increasing, but also the challenge is how we can satisfy these needs, and also the patient will be satisfied, and also the cost to have access to the drug also will be less expensive. So this means that we have to think in the innovative approach, how we will make this happen. We think that as the pharma industry, we cannot achieve this goal alone. This is why for us, innovation is key. So working with the digital technology, all people working in this ecosystem are welcome to make this happen. So this said, so currently, this is the normal patient journey. So the diagnostic, treatment, challenge to have access to the treatment, because sometimes the cost or the distance between the patient and the health facilities, and also how this treatment will work depending on the patient. So that during the different trial we did across the world in oncology, Roche is one of the leading pharma industry in oncology. We noted that for cancer, some cancer are common in Western countries, like breast cancer. When you take breast cancer, there are different types of breast cancer. HER2, triple negative breast cancer, and also hormonal breast cancer. So for us, we noted that in Europe, the HER2 are more common than in Africa. And in most of our trial, we have less Black people enrolled in our trial. So that, this is why we think that the patient will react differently regarding one treatment in comparison to another treatment. So we think that in the future, I cannot say 10 years, but in near future, so this is how the patient journey will look like. Early and accurate diagnosis and plan a personal treatment. This means that the treatment that will work for this patient will not be the same. You know, for today, we have what we call the guidelines. This means that if you have the breast cancer HR2 positive, we have to use this first line. After that, you move to the second line. But in the near future, this will not work. This means that for these patients, this is the right treatment for that patient. For us at Roche, personal healthcare means that right treatment, right patient at the right time. So this is for us. So we are trying to put together our whole energy. So in Roche, we have what we call Roche Pharma. They are working in the development of the drugs. Roche Diagnostics for the diagnosis of the disease. And also we have a different platform, Foundation Medicine, FMI, Flatiron, Navify, so that we think that bring all these internal institutions together in what we call One Roche, we think that we can shape the ecosystem and achieve this personal healthcare soon. So for that, we have to work on different pillars, the ecosystem policy, the regulatory, building the capacity, access to treatments at a very affordable cost, access to a diagnosis. Early diagnostic also is very key. We know that when the cancer is diagnosed at early stage, the guérison, the guérison is accessible. Data policy and also how we can work on the real world data, which are the different types we have. So in Africa, we are all committed on that. This is a mapping we are doing in French-speaking Sub-Saharan Africa. This is FSSA and Portugal-speaking Africa. So that this is where we are. We are at a very early stage. But we think that in the partnership with different institutions, we can move fast and also achieve this goal quickly. So in terms of initiative, so these are some initiatives we have started in terms of making the personal healthcare a reality in Africa. So that the big one is working on the genomic. So as I'm saying, I was saying that in the whole trial, not only for Roche, but when you take all the trial performed in the oncology, you will see that the African genome is less than 2%. So this means that something has to be done. So we bring together what we call the Africa Genomic Project. Is a huge project to have the profile of at least 50% of African people. So that we have started with the profiling of the triple negative breast cancer patients. This is in collaboration with Pasteur Institute. 4 countries are involved in this project: Congo, Ivory Coast, Senegal, and Tunisia. We had the review of the protocol, and we think that the first quarter of next year we will start this huge program. We are also working with the Swiss tropical public health, with the Centre de Research Suisse in Ivory Coast, to also to grant the reagents to perform a study on to have, you know, the profile of the metabolic hypertension and diabetes patient in Ivory Coast to understand. And in South Africa, this is a huge coalition involving the other pharma industry like GSK, Pfizer, AstraZeneca, and the University of KwaZulu-Natal to profile 100,000 patients in South Africa to have the genetic profile. This is some initiative. They have the others, so because of the time, I have just summarized the 3. And also we have signed an agreement with African Research foundations to support the emergence of the African leaders. So we will provide a grant to free African young researcher, 9 month grants that will go in the very renowned lab across the world to have 9 month immersion and to develop their skill in research. After that, they will come back in Africa and to be integrated on all these projects we are running across Africa. So these are some initiatives so that through the partnership we are built with the different MOH and the government, so that in South Africa we are working in the building capacity and skills. In Ivory Coast, this is a basket deal we have signed with the government of Ivory Coast to make accessible our innovative treatment, but also to work to shape our ecosystem, to develop the human resources, so that in Q1 2024, we will run our clinical trial in Cote d'Ivoire. Free health center has been standardized, to be at the very high level, so that the clinical trial can be done there, so that the patients, the Black population living in Africa can be involved in this kind of trial. So that also we are working to tackle some infectious disease like HIV, TB, and also HPV. We are part of the coalition for the elimination of the cervical cancer by 2030. So that these are some initiatives. And as I said, we Roche, we are open to collaborate with the young researcher across Africa, also to develop a partnership to make available the innovative solution we are bringing. Regarding the personal health care alone, we can't, but together we can make it happen soon. Thank you very much for your attention.
Thank you, Dr. Gino, for sharing your project from Roche. Now we have finished with our four speakers. So if you have a question, you have a little time. It's very late. So you have the floor. If you have some questions for one of our speakers.
Thank you. Thank you so much. My name is Stella. Great job to all the speakers. I really knew.
So it goes far beyond. The answer go far beyond personalized medicine. The European people of the West, America, Europe, they can work together. They can sit together. They have the ability to sit together, set common objective, plan their business, find a way to tackle their objective, and
l'Union européenne. Est-ce que vous ne pensez pas que vous devez peut-être nous asseoir dans le cadre de ce que vient de dire Badara, essayer de potentialiser un peu ce que vous faites dans les pays? Voilà.
OK. OK. Merci beaucoup. To your question regarding the accessibility of our medicine, I can say that for us at Roche, innovation means that how we can act differently to make available our innovation accessible to the patient in need. This is why we put together what we call our ambition for Africa. So I'm gonna give an example of Ivory Coast. In 2019, we agreed with the government of Ivory Coast on what we call the basket deal. The basket deal is an agreement where Roche and the government of Ivory Coast, we sign an MOU. In this MOU, the government of Ivory Coast will bring money. I think that is almost 5 million CHF, and we Roche will bring our innovation, and in addition to that, we will develop the human resources. We will build the ecosystem, and also we will bring our drug at zero cost for the patient. When we started this program in 2019, in Ivory Coast, we have, I can say, 4 medical oncologists. But today, with this agreement, we have 14, so that we are paying the fees for these medical oncologists. We are working now to open the cancer treatment center in the CHU de Bouaké. Bouaké is the second city of Ivory Coast. We are also improving the diagnostic capacity. Last month, during the Pink October, the head of our Center of Reference in terms of treatment of cancer, which is CNRO, this is, I will quote what she said. She said that because of this agreement between Roche and the government of Ivory Coast, they increase, you know, the life, I cannot say the life, but they decrease the mortality of breast cancer by 25%, because we are working on the disease awareness. So the women are coming earlier to seek care. So this is a kind of partnership we are trying to build across Africa. Currently, we are in the discussion of the MOU in Senegal. We hope that we will sign that agreement to come to the personal health care, so that we think that as I said, we alone, we cannot, because we do not have the whole solution to treat cancer. This is why we are working in the coalition with the other pharma company. We are working with the universities. We are working with the center of research like Pasteur Institute. Pasteur Institute is very strong in Francophone countries. So we are working with them to bring the solution for Africa. So we think that together we can reduce the cost. Today I can say that our drug is at zero cost for cancer patient in Ivory Coast. So I'm going to take an example for the hemophilia A patient. They are receiving our drug for free, but the same drug in US costs 5 million euro. You see, so that this is because of the agreement we signed with the MOH in Ivory Coast. In Senegal, we are working on it. So professeur, donc c'est ça pour vous dire que Roche, nous sommes ouverts à tout type de collaboration. On a initié les discussions avec l'IRSF. Comme je le disais, c'est dans des partenariats qu'on peut trouver des solutions aux problèmes de l'Afrique. Et pour ce type de partenariat, nous sommes engagés, d'où notre présence ici. Et on pense qu'ensemble, avec l'IRSF, avec toutes les universités, et l'avantage du Sénégal, c'est qu'il y a beaucoup de ressources humaines très qualifiées, très compétentes. Donc, on pense qu'ensemble, on pourra travailler pour mettre en place des solutions innovantes pour les populations sénégalaises, pour ne pas dire au-delà du Sénégal.
So much. So, Badara, you can deal with it. Thank you so much. We have finished with our session. And thanks you for your participation. And have a good night.